Hello, my name is Patrick Quinn. I have a PhD and HCLD qualifications. I'm Vice President of Research and Development at Sage IVF, which is a Cooper surgical company. My landmark paper in 1985 was for the development of HTF medium, and from 1996 I've been in the commercial field of developing IVF culture media in the assisted reproductive technology field. The company was purchased by Cooper Surgical in 1999, and we have developed and refined our culture media for all the aspects of human IVF work since that time. We have a sequential series of culture media for all stages of pre-implantation development of human embryos, from collection of the oocytes from the ovaries through to development of the embryos through to the blastocyst stage. These series of media are the fertilization media, cleavage media for the first 3 days of development from day 1 through day 3, and blastocyst media for continued development from day 3 at the 8-cell stage to day 5 and 6 at the blastocyst stage. These series of media come in two forms: one without protein, which is the Quinn's Advantage series. Their catalogue numbers are 1020, 1026 and 1029 for fertilization, cleavage and blastocyst medium. And then we also provide these media in a more convenient form with the protein already included. These are the Protein Plus series of fertilization, cleavage and blastocyst medium. Catalogue numbers 1520, 1526 and 1529 respectively. As well as our culture media series, we have other media products for other aspects of handling embryos, oocytes and spermatozoa outside of the CO2 environment in a culture incubator. These are procedures such as cryopreservation, the ICSI procedure, embryo biopsy for pre-implantation genetic diagnosis. We also have media products for handling spermatozoa, for washing the spermatozoa and for cryopreservation of spermatozoa. Our culture media are based on the principle of imitation, where we imitate the chemical environment that is found in the human female reproductive tract. I have found over the years that two of the most critical aspects of using our culture media are the correct pH of the media and to monitor and maintain the correct temperature of the media during the culture period. It's very important to measure the actual pH of each lot of media as you receive it in your laboratory, and not to rely on the CO2 concentration or readout of CO2 concentration on your incubator. It is essential therefore to have a pH meter and to regularly check the pH of your media. When measuring the pH and temperature, it's important of course to use validated instruments for these processes and to look for any differences in temperature gradients within the incubator or on the microscope stage or any other place where the embryos and gametes are utilized. These stages of embryo development, from oocyte through to the blastocyst stage, are very sensitive to any dramatic changes in their culture media environment, and we believe it is important to culture the embryos in one line of media from one particular company. So for the Sage series of media, we believe it is important to transfer embryos that have been fertilized in Sage fertilization medium to the Sage cleavage media, and then finally to the Sage blastocyst media. This gives the greatest opportunity for patients to obtain a healthy baby at the end of the whole IVF procedure. So over the 25 years since the development of the original HTF media, I've had a lot of opportunity to make improvements in the culture media, and we believe that our series of media provide the best opportunity to get the highest rates of development and to produce the most viable embryos. And definitely this gives a greater opportunity to do elective single embryo transfer and to reduce the incidence of multiple gestations.